Howdy folks, as we jump into subtask 3, which is going to be found on pages 4 and 5 of the Learning Targets homework, I'm just going to encourage you that we're going to be getting into some new information here. And so you might want to use the notes that are up on Google Classroom because those are going to be fully filled out. They're going to have all the information there for you to follow along. I would also say that if you find yourself confused, you might want to note that and make sure to reach out to me or at least go online and find some other resources to help you clarify that. The responsibility is on you to make sure that you're getting the help that you need. That's part of this independent work. Now, subtask 3 is actually going to be all about the genetic code and what happens to it. So this is not in your notes, but it is important to know that the genetic code is universal. A's, T's, G's, and C's. That is going to be what every living thing on Earth does. So whether you are a tree or a plant, whether you're a chimpanzee, whether you're any animal, whether you're a fungus, whether you are a jellyfish, whether you're even a bacteria, all of these things use DNA and they use A's, T's, G's, and C's. So the genetic code is completely universal. In fact, it's so universal that you can actually take genes out of one organism, put them into another organism, and they still work. In this case, the gene that is being transplanted is a glowing protein gene. And if you take that glowing protein gene and put it into cells, the cells glow. If you put it into frogs, the frogs glow. If you put it into mice, the mice glow. Chimpanzees, bugs, even cats glow. So the genetic code is so universal, you can actually take genes from one thing and put them in another, and they still function because of the central dogma. The DNA is read, that makes RNA. RNA is then used by a ribosome to create a protein, in this case, a protein that glows. So the genetic code is completely universal. And with that, I would just like to point out a few of the features of the genetic code. This is going to be where you describe the genetic code in detail. This is the first part of subtask 3. So you're going to want to note that it is universal. You are going to want to note that it is made up of a sequence of bases, a sequence that is written A's, T's, G's, and C's. We can also note that there's a direction to the genetic code. You read it from 5 to 3. So wherever carbon 5 is, the direction towards carbon 3, that is the direction that you read that genetic code. Now, each strand of DNA is made up of millions of base pairs, millions and millions of letters of genetic code. And in the total human genome, 23 chromosomes, there's actually a few billion base pairs. Now, if you have one half of the DNA, you should be able to write in the other half. We've practiced this a little bit in class, so you're going to be just doing the base pairing. So for an A, there's a T. For a G, there's a C. Now, the next thing to note here is that sometimes the genetic code changes. Sometimes you have what's called a mutation. And a mutation would be any change to the genetic code, and this could happen for a couple of reasons. Maybe you're doing DNA replication. That's what we're going to talk about in the last half of this unit, where you actually take the DNA and you create an identical copy of it. Think of creating that identical copy like you writing down some notes in class. Every so often, I'm sure, you make a mistake. The word on the board is just a little different than the word you wrote down. Maybe you changed a letter, maybe you changed a whole word, and you didn't do it on purpose. That can happen during DNA replication, where there's a mistake made and then the genetic code has been changed, but not because it was trying to change, just because there was a random error in the copying process. There are also mutagens, carcinogens, things that can actively change the DNA of organisms, and those would be cancer-causing agents, things that can cause mutations to genetic code. So for instance, cigarette smoke, ultraviolet light, certain chemicals, those are dangerous because they can alter your DNA, changing parts or even whole portions of chromosomes. So there are a few different kinds of mutation or changes to the genetic code. And the analogy that I want to use are these little sentences, and these sentences are made out of 3-letter words. The 3-letter words are important because the 3-letter words represent a codon, or 3 letters of the genetic code. Genetic code is always written in codons, little 3-letter words, so I'm using 3-letter words of the English language here as an analogy. You'll notice at the beginning, there's always a start codon. And that was the codon during the central dogma lab that everybody had first. AUG in the RNA was the first codon always. So there's always a start codon. There's also an ending or a stop codon. Everybody should have had a sequence of bases that basically said, don't put an amino acid on, just stop. So we're always going to have that too. And in the middle, we've got a few codons. In this case, the man saw the car hit the dog. All of those are 3-letter words. So what would happen if we did a mutation to that? Well, the first mutation that we'll take a look at is a point mutation. And if you do a point mutation, what you do is you change one nucleotide. You just swap it out for another one. So in this case, we're going to change the base R into a T. Now I know that there is no R in DNA. This is an analogy. But in the genetic code, this would be taking an A, T, G, or C and swapping it out for a different base. That changes the codon. So now it says, the man saw the cat hit the dog. Has a little bit of a different meaning. This one could be significant if the codon that you change ends up putting on a different amino acid. But as you may remember from the chart, there are actually multiple ways to code for one single amino acid. It's possible that depending upon which thing you change, you might not actually change the amino acid you put on at all, and then the protein might be the exact same shape and function the exact same way. So point mutations, they might be significant. They might also not matter at all. The next kind of mutation is called a deletion. Now, this is where you actually remove a letter from the genetic code. You could actually remove anything from a single letter to an entire gene. You might remove one or more letters. Now, in this case, what I did is I removed 3 letters, which is lucky because that means that we're not going to cause a frame shift. A frame shift is the last one we're going to talk about. So for right now, as long as I'm removing multiples of 3 bases, in this case, I've removed just 3, the 3 that would be right here, car. All the words are still the same, we're just missing a word. The man saw the hit the dog. So this is a pretty significant change. If this was the case, there's one amino acid that we are not adding to our protein anymore. That might have a varying degree of effects. Anything from not very significant to very significant. The next kind of mutation is called an insertion, and this is the opposite. That's where you add some DNA, anything from a single base up to an entire gene. But the effect is going to be roughly the same. In this case, I inserted 3 bases, and because I did that, because I added a multiple of 3, all of the other words are still the same. So now we have the man saw the big car hit the dog. Again, this would mean that we added a new amino acid in that might have some ramifications for the protein. But the real one that I wanted to get to is the frame shift mutation. Now the frame shift mutation is a version of a deletion, but what you've done is you've removed not 3 or a multiple of 3 bases. And what that means is that every letter of the genetic code is going to slide over a spot. And what that means is that every single codon after the deletion is different. So what we can see now is there was a frame shift right here with the C. We no longer have the C, and if you take the C, the next word you have, if you look at just the next 3 letters, is A-R-H. That's not a word. And then we have I-T-T, and then H-E-D. You see how that happens? If you keep reading 3 at a time, but you shift them over either 1 or 2 bases, every single word changes. So now our sentence is, the man saw the arch-itit head ogi, and then our end codon isn't even an end codon anymore, it's just nda. So we might not actually have a stop signal, and every single codon is different now. That means that we could have a different set of amino acids at every single spot for the rest of the protein. This one is very dramatic. This one is the most dramatic and will have the most effects on the genetic code if you have what is called a frame shift. And that's where you're shifting the frame of the codons, again, because you have removed a multiple of bases that is not a multiple of 3. Deletions might have an effect, but a frame shift deletion, that's going to be a really big deal for the organism. All right, so With all of those in place, some mutations are definitely more significant than others. It really just depends on how much of a change to the genetic code there is.